Hello everyone, welcome back to sqadia.com. Today we are back with another video of Staphylococcus Infection Case Study for better understanding, so you will be able to identify not only the type of gram-positive cocci involved, but also able to distinguish between different species of Staphylococcus. Now, without wasting time, let's move toward the case study. A patient named Ronald, who is a 34-year-old man and an athlete, whose medical history was significant only for chronic knee injuries over the past 12 years. Ronald's initial surgery appeared to have gone well, and he was discharged after several hours, as is usual for this type of surgery. However, he reported to the emergency room 2 days ago, with fever, pain, and swelling of the knees. Blood cultures were collected and a sample of the exudate from his knee was also submitted to the laboratory for gram staining of the exudate that yielded numerous neutrophils, rare squamous epithelial cells, and many gram-positive cocci in clusters. And 2 lab tests were performed that shows that the organism is coagulase positive and catalase positive. Now, based on the case presented, can you identify the organism? What additional tests you need to perform? And which drug should be given perioperatively, so that surgical wound infection would not have been occurred in Ronald? Not able to answer these questions? Don't worry, we will solve the case together. Try to recall our lecture on Staphylococcus, as it is mentioned in the case that gram-positive cocci were present in the blood culture. So we have studied in our previous video on Staphylococcus that there are 2 medically important genera of gram-positive cocci, and that are Staphylococcus and Streptococcus. Both Staphylococci and Streptococci are gram-positive cocci, but they are distinguished by 2 main criteria, and that are Staphylococci appear in grape-like clusters, whereas Streptococci are in chains. Staphylococci produce catalase, whereas Streptococci do not produce catalase. Case presents that the isolated organism is catalase positive and are present in clusters. So among Staphylococci and Streptococci, Staphylococci should be present, and we already know that catalase is an enzyme that degrades hydrogen peroxide into water and oxygen. But we have also discussed that Staphylococcus consists of 3 species, and that are Staphylococcus aureus, Staphylococcus epidermidis, and Staphylococcus saprophyticus, and the tests were also discussed in our previous video that are performed on Staphylococcus. These tests are coagulase, color of colony, and hemolysin production. Staphylococcus aureus is coagulase positive, but Staphylococcus epidermidis and Staphylococcus saprophyticus are often referred to as coagulase negative staphylococci. Staphylococcus aureus produces a carotenoid pigment called staphyloxanthin, which imparts a golden color to its colonies. This pigment enhances the pathogenicity of the organism. Staphylococcus epidermidis does not synthesize this pigment and produces white colonies. Staphylococcus saprophyticus also does not synthesize this pigment and produces white colonies. Staphylococcus aureus produces hemolysin, but whereas Staphylococcus epidermidis and Staphylococcus saprophyticus do not. And it is already mentioned in the case that the organism is coagulase positive. So, according to presented case, the organism must be the Staphylococcus aureus. Before proceeding further, have you ever thought how these tests look like? No. So let me show you. This is a coagulase test. Left test tube shows positive coagulase test, as we have discussed that coagulase test is positive in case of Staphylococcus aureus. But right-sided test tube shows negative coagulase test as in case of Staphylococcus epidermidis and Staphylococcus saprophyticus. This Petri dish shows golden colonies, and we have studied that Staphylococcus aureus produces golden colonies due to carotenoid pigment called as staphyloxanthin, while this Petri dish shows white colonies in case of Staphylococcus epidermidis and Staphylococcus saprophyticus. We have also discussed about catalase. This is positive catalase test, And we also know that it occurs in case of Staphylococcus. But catalase test is negative in case of Streptococcus. Is there any additional test you need to perform for Staphylococcus aureus? Yes or no? The answer is yes. So what's that test called? This test is called as mannitol fermentation test because Staphylococcus aureus is able to ferment mannitol. Let's move into bit detail. What is mannitol fermentation test? Well, in this test a media is used. It is a selective and differential media for isolation of Staphylococcus aureus, which contains high concentration of sodium chloride, and that is 7.5%, because Staphylococcus aureus can tolerate high saline levels. Well, in this test, Staphylococcus aureus can ferment mannitol, colonies will appear yellow, but the Staphylococcus epidermidis that cannot ferment mannitol, in this case colonies will appear pinkish. This media is called as differential and selective. But why? Well, it's differential because Staphylococcus aureus can ferment mannitol and give diffuse yellow color, while it is called as selective because it contains high salt concentration, in which only Staphylococcus aureus can grow. Now, which drug should be given perioperatively so that surgical wound infection would not have been occurred in Ronald? Try to recall the lecture on Staphylococcus aureus. Can you remember the drug that should be given perioperatively to prevent Staphylococcus aureus surgical wound infections? So the drug of choice in this case is cefazolin that should be given to patient perioperatively to prevent surgical wound infection. Wait, hold on, let me ask you another question. What if the organism was catalase positive but coagulase negative? Obviously, it must not be the Staphylococcus aureus. But then which organism it should be? Recall remaining two species of Staphylococcus, and that were Staphylococcus epidermidis and Staphylococcus saprophyticus. Both are catalase positive, but coagulase negative. These are two species of Staphylococcus. Now, how will you be able to differentiate them? The two coagulase negative Staphylococci are distinguished by their reaction to the antibiotic called as novobiocin, because Staphylococcus epidermidis is sensitive to novobiocin, whereas Staphylococcus saprophyticus is resistant to novobiocin. That was all about today's video. Stay tuned to sqadia.com for more precise and knowledge stuff videos.